Good evening everyone, I'm Stephen Chu and I would like to discuss about my favorite topic from biology, which is known as enzymes. Before I proceed discussing about enzymes, I would like to first mention the term called catalyst. What is a catalyst when it comes to your mind? A catalyst is a substance that speeds up the rate of reaction, thus remain unchanged throughout the reaction. Therefore, an enzyme itself is considered a protein that acts as a biological catalyst that speeds up metabolic reactions, thus remain unchanged throughout the whole reaction. And the reason why enzyme is sustainable to our lives is that most metabolic reactions are controlled by enzymes. Therefore, if there are no enzymes, then metabolic reactions will be slower. And now I would like to proceed to the enzymatic action, where it describes the lock and key mechanism that also illustrates how enzyme and substrate operates. So I would like to start off with the enzyme here. And let's say this enzyme is an amylase, as it's just because it's a common carbohydrate that I've heard of. And let's say the substrate here will be starch. And since most enzymes are specific, we should ensure that the starch has a complementary shape that could bind onto the active site of the enzyme. And once it binds into the active site of the enzyme, it will form what's called the enzyme substrate complex here. As it bound into the active site, the amylase will be responsible for breaking down the starch into two molecules of maltose, and it's maltose because it's carbohydrates. So let's say here, these two molecules of maltose will be the end product following the breakdown reaction. And now I would like to discuss about the other properties of enzymes that's significant for you guys to understand. And we know that most enzymes work best at an optimum temperature and pH. But first, I would like to start with how temperature affects the enzymatic activity here as I draw the graph. So let's say this is the graph, and let's say the optimum temperature here will be 37 degrees Celsius. And now we know that as we increase the temperature, we always consider that there will be high rate of chemical reaction, and this is because when you increase the temperature, you apply more heat and kinetic energy, and due to more kinetic energy, it will cause frequent collisions between molecules, and due to frequent collisions, that's causing the chemical reactions to operate rapidly. However, in terms of enzymes, if you increase the temperature above its optimum, that will lead to what is called the denaturation of enzyme. This when enzymes lose its shape or structure, thus the substrate will not be able to bind to its active site, causing no reaction. And this because when you increase the temperature, same thing as I mentioned earlier, you cause frequent collisions taking place. And these frequent collisions are the ones that disrupt the bond that's holding the enzymatic structure, because we know at first earlier that enzymes are proteins. So these collisions disrupt the bonds that's holding the protein structure, and that's what's causing denaturation. And now I'd like to discuss about how pH affects enzymatic activity, as I'm going to draw here. So I'm going to show you guys the acidic enzyme and the neutral enzyme, which I'm going to draw here. Let's say for neutral its pH is 7, and for acidic its pH is 2. If you either increase or decrease the pH for neutral enzymes, that will cause denaturation. Same applies to the acidic ones, where if you increase the pH above 2, that will also lead to denaturation. And this is because an extremely amount of pH would disrupt the bonds that's holding the enzymatic structure, thus causing denaturation to take place. Thank you so much and have a nice day.